Welcome back again, dear students. Today, our topic is anatomy of dicot root. The primary structure of a typical dicot root under microscope shows following structural details. Starting from periphery, we have the outermost layer, which is known as epiblema or pelliferous layer. Simply, you can say epidermis in case of roots is known as epiblema or pelliferous layer because it possesses extensions in the form of unicellular root hairs. So let's move on with what is epiblema. It is a single outermost layer of thin-walled cells, and some of these cells have an outward extension in the form of unicellular root hairs. The primary function of these root hairs is absorption of water and mineral salts from the soil. This point is very much important. There is no cuticle on the outside of these cells. Okay, cuticle is absent in case of roots. Then cortex. Cortex lies below the epiblema. This region is multilayered and is made up of parenchyma cells. Then endodermis. It is a single circular layer of barrel-shaped cells, and these barrel-shaped cells are compactly placed, leaving no intercellular spaces between them. Intercellular spaces are absent between the barrel-shaped endodermal cells. The tangential as well as radial walls of these endodermal cells, they possess Casparian strips, which are composed of a waxy substance which is water impermeable, and that waxy substance is known as suberin. Okay, so Casparian strips which are present on radial and tangential walls of the endodermal cells of roots, they are composed of water impermeable suberin. Then pericycle. It consists of a few layers of thick-walled parenchymatous cells, and this pericycle is very much important in case of roots because it forms the seat of origin of lateral roots. This is the main root. Okay, the lateral roots that form, they actually originate from the cells of the pericycle. Okay, and one more important function of pericycle in roots is it is involved in secondary growth. That means the individual pericycle cells, they become meristematic, they form cambium, and these cambial cells form secondary xylem and secondary phloem. Then the vascular bundles. The vascular bundles in case of roots are radial, they are X-arc. Okay, let's make it more clear. This is the protoxylem group, this is another protoxylem group, another protoxylem group. Okay, and in between these protoxylem groups, you have phloem. That means in case of dicot roots, the xylem, it alternates with phloem. This is xylem. Okay, this is xylem, it alternates with phloem. Okay, that means they are placed along different radii. The xylem and phloem are placed along different radii. And one more important feature about the xylem of dicot roots is, or you can say general roots, is it is X-arc. X-arc means this is protoxylem, okay, which is located towards periphery. The metaxylem will be placed towards the center of the stem. The metaxylem will be placed towards the center of the stem, so that you can say the direction of development is centripetal. Okay, this is protoxylem here, the first form is xylem. This is the metaxylem, the xylem which is formed later on and is placed near the center of the stem. So you can simply say it is X-arc. And this line is very much important. The vascular bundles in case of roots, dicot roots specifically, it is limited to 2 to 6. It cannot go beyond 6. Then the conjunctive tissue. The conjunctive tissue consists of parenchyma cells which lie between xylem and phloem. This is protoxylem. This is phloem. This tissue is known as conjunctive tissue, and it is made up of parenchyma cells. Then finally the pith. The pith is either poorly developed or absent. You have to remember that the pith is either poorly developed in case of dicot roots, or it is altogether absent. And in some cases, in some rare cases, if it is present, it is made up of parenchyma cells. But by and large, you have to remember that pith is absent. Now let's have a look on this uh, slide so as to know the internal structural details of the dicot roots. The outermost layer is known as pelliferous layer because it possesses root hairs. The inner to this we have this uniform cortex, okay, made up of parenchyma cells, and the innermost layer of this cortex is endodermis. The endodermal cells possess Casparian strips on their radial and tangential walls, which is made up of suberin. Then we have the pericycle, which is multi-layered here. Okay, here it is only single layer, but you have to remember that in the image it is they have just made it single layer for the sake of convenience. But you have to remember that the pericycle in case of dicot roots it is made up of many layers. Then this point is very much important. Here you can easily see the vascular bundles. Okay, this is one protoxylem group, another protoxylem group. This is another protoxylem group and another protoxylem group. Okay, and in between these protoxylem groups we have here phloem, another phloem patch, another phloem patch, and this is another phloem patch. Okay, I have told you that the xylem in case of dicot roots or generally roots, it is X-arc. X-arc means the protoxylem is located towards periphery, and this central part, okay, the central vessels, it is metaxylem. That means the xylem which forms later on. It is located towards the center, and the direction of development of xylem is therefore centripetal. Okay, I think this is all about the anatomy of dicot roots. I hope you all understand this. Hopefully in the next class we are going to discuss the anatomy of monocot roots. Till then, stay blessed and thank you all.